collections myself, and I'll take a stab at the square or rectangular pupil. So the goat would be a great example if you've ever looked at a picture of a goat pupil. If you want to find a rectangular pupil, a good place to look is among hooved animals that live on the prairie, because their preferred field of view is on the along the horizon. I hadn't thought of the octopus. My answer to the question, which I thought probably people nobody else would have, was the great white shark, which has a horizontally elongated pupil, and I don't know why. So I'm going to screen share here and call up my presentation. Can everybody see that, and can you see my cursor?
Yes.
So I am going to talk about the eye collection at the Comparative Ocular Pathology Laboratory of Wisconsin. I am a veterinarian, and I'm a veterinary pathologist. I did graduate from the University of Minnesota in 1972, and then I began a pathology residency at the veterinary school at the University of Pennsylvania in 1974. Early in my training program, I was mentored by a man who was a neuropathologist. He specialized in neuropathology. And although he wasn't the person for whom I give credit to learning pathology, I did come to the conclusion that a good way to do a career in pathology was to choose a organ system specialty and make that the core of what you do. It's by no means the only avenue to do a career in veterinary pathology, but it's what I chose to do. Originally, I thought that endocrine pathology would be a good match, and I teamed up with an endocrinologist, but he left, and I was invited to do pathology rounds with the ophthalmology trainees by a man named Gus Aguirre. And Gus Aguirre is a highly renowned, respected veterinary ophthalmologist and vision scientist. So he became my mentor in ophthalmology, and that's been it since then. Madison, Wisconsin is my hometown, which is one of the reasons why I was excited to leave the University of Pennsylvania and come back to Wisconsin, and that happened in 1983. As a contingency to hiring me, I asked that I be allowed to have a mail-in, not so much a mail order, but a mail-in eye pathology lab, which would have a fee-for-service funding operation, and then I would have control over the fee-for-service, the money that came in for the fee-for-service funding, and so that's how that happened. It was a contingency of my original employment at the University of Wisconsin. So I wasn't hired as an eye pathologist. I was hired as a general pathologist, and really wasn't until my children left the house to go to college, one after the other. And my wife at that time was a 8th grade science teacher and also a perfectionist. And if you put those two things together, you realize that she was working all the time. So I realized that I didn't have as many home responsibilities as before, and I could work all the time too, and I could throw the doors open and try and increase my ocular pathology service. So I did that, and it wasn't too long until I was able to afford people to help out with the lab, and was able to make that my job rather than just a interesting hobby. One of the things we had to do is we had to pay the salary of someone who would replace me as on the diagnostic team at the veterinary school. So this is an example. This is the type of thing that's our end product. These are microscope slides from tissues from a dog. It looks to me, I don't remember, but it looks to me like it has some sort of inflammatory disease. and we're looking at a number of different stains to help us identify what the pathology is. Based on the fact that there's a green stain here, I'm guessing that at least we thought there might have been a fungal disease. So just a little bit about the COPLOW history, some of which I've said already. It was 1983 when I left the University of Pennsylvania, already interested in eyes, and established the Comparative Ocular Pathology Lab of Wisconsin, which we call COPLOW, as a fee-for-service lab. By 1995, already my kids had gone off to college, and I was working full-time, trying to do both the job I was hired to do and also to run this expanding eye pathology program. At that time, we initiated a fellowship, which is a one-year training program attracting veterinarians who either have an interest in getting into a ophthalmology training program or a pathology training program. And since the ophthalmology training programs are much harder to get into, most of our fellows were people who were interested in ophthalmology. They had to be trained in pathology, but they were very, very highly motivated individuals. And it wasn't too long after that when we were able to hire a lab manager. All of these advancements depended upon money that we could afford from the fee-for-service. So the more cases we get in, the more money we have in the lab, and the more we can hire. But also, it means that the more we work in order to keep up. So in 2014, I retired, and Dr. Leandro Teixeira, who had been working with me for about 5 years, oops, don't like that. I asked him before I retired. I couldn't imagine going on without him, and I couldn't afford to keep him on at a salary he deserved. So and the funding for him dried up. So I asked him if I retired, would he apply for the job, and he enthusiastically said yes. So that's what happened. He took over in 2014 as the director of the lab. Gillian Shaw is a veterinarian who did both her pathology training and her PhD at the Wilmer in Baltimore, but she her PhD training was in basic science, retinal disease, but she calculated her interests, and she was more interested in being part of a diagnostic service than continuing on in the type of research that she had done at Wilmer. And then in 2020, we hired another already trained pathologist, Ryan Taylor, who had done his training, both his veterinary degree and his pathology training at the University of Mississippi. And then just a few weeks ago, we hired Kelly Brakel, who is also a board certified pathologist and just beginning her training as a specialist in ocular pathology at our lab. So we have 4 full-time dedicated eye pathologists, and then myself as a volunteer. And I continue to do work to the benefit of the lab. I work with the database, and I work with the images, and I help out particularly with the comparative anatomy. I love my passion in my retirement is comparative anatomy of the eye. So this, in a nutshell, kind of describes COPLOW's mission. As with most academic programs, it has or at least programs in a veterinary hospital setting. It has 3 missions. One is to provide a diagnostic service, which would be on the top. So our clients are mostly board certified or specialist trained veterinary ophthalmologists. And last year, the pandemic is veterinary medicine is one of those businesses that benefited from the pandemic. Everybody wanted a companion animal when they had to home isolate. So businesses among veterinary ophthalmologists around the country skyrocketed, and for that reason, our businesses skyrocketed. So we had a record-setting more than 7,700 accessions in 2021. And then we have a continuing education program. The fellowship program that I mentioned briefly earlier is now in its 22nd year. So every year we've had a new individual who trained with us for 1, sometimes 2 years. And then we've just started to hire clinical instructors, and that's where Kelsey Brakel and some of the other people come into our program, already trained as pathologists, and in some cases already trained as eye pathologists. And then we have a very busy visitor program. People can sign up to visit our program, usually for 2 weeks, but some people come for shorter periods of time. Some people come for several months, and some people come on sabbatical leave for half a year or even more. So that's another program that we are proud of in terms of our continuing education. And then, of course, all of us do presentations. We wrote a textbook a few years ago, and so we have a continuing mission to become a gold standard or a go-to laboratory for continuing education in veterinary ocular pathology. And in the research that we do, the core of our research is investigating the natural history of animal eye disease, which means that we receive specimens from animals that have eye disease, and then we use our pathology training skill to investigate the morphologic anatomy of the eye disease and put together observations that help to better define the nature of the eye disease, either the cause or the separating out different types of eye disease. We do an awful lot of that. I, probably more so than the others in the lab, although the others are interested as well, are interested in comparative anatomy. And we also do participate in some research programs to elucidate some interesting features of ocular structures in different types of animals. We are a resource for collaborations with more bench-related vision scientists. So let's say a scientist is working on an experiment involving the eye, involving some mice or something like that. And if the eyes become available for evaluation by pathology, we will do those studies. And then we also, through the years, we've also become a center for collaboration with industries that are working on drug development, particularly working on drug development for ocular pharmaceuticals. But we will also come into play if a drug company, for instance, is working on drug development in something with no expected ocular benefits, but they run across an ocular toxicity. So we might be called in to help evaluate that type of thing as well. This is Dr. Teixeira, and this is a picture of our collection. The brownish boxes below basically houses all of our microscope slides, and I showed you some examples of microscope slides. And the box that he's opening contains paraffin blocks. This particular segment of the collection contains the paraffin blocks for extra large eyes, for instance, horse or sea lion or tiger or something like that. Extra large blocks would be in those. So our slide collection, microscope slides, fits nicely into a very confined space, but the block collection has really spread all over it can't be housed in the room where our lab is. It's spread in nooks and crannies all around the veterinary school, and we're going to have to come to grips with that sometime soon. We're going to have to probably hire some curating service to hold on to our blocks in such a way that we can have access to them when we need them. So this is our bread and butter. This is what we get. This happens to be a horse eye that's sent in a plastic container and fixed in formaldehyde. Almost all the eyes we get are already fixed in formaldehyde. They are eyes that come from veterinary ophthalmologists who, for one reason or another, decided that it's medically appropriate to remove the eye from an animal because of eye disease. Or about 2/3 of our submissions are actual globes, actual eyeballs, and the other third are tissues from around the eye, like an eyelid tumor or an eyelid inflammatory disease or something from the orbit, some other tissue that would come into contact with a veterinary ophthalmologist. This particular specimen is a horse eye. It's a big eye, and you can see that it's got some sort of corneal disease that's causing the cornea to turn a orangish yellow. This is another look at what we save. On the right is a microscope slide, probably from a dog or a cat eye with a pigmented neoplasm in the eye. So those are this slide would be housed in that housing compartment that I showed you with Dr. Teixeira. And then in the middle is a paraffin block. That's the actual tissue from the eye that's been completely embedded with paraffin, and it's from the paraffin tissue. It's from the paraffin block that our technologists, our histotechs, are able to make extremely thin, about a 5-micron section of that tissue, and then mount that on a microscope slide, and then stain it with whatever is appropriate stain. The most common stain we use is this pink and purple stain that you see on the right, and it's a combination of the stain hematoxylin and eosin, which I'm not going to get into those things. On the left is a little plastic container. When we trim the eye, we actually put it on a block, and we use an overly large razor blade to cut right down the middle in the vertical plane, and then put a section of that in this little plastic container that then houses it as it goes through the histology lab, eventually ending up in paraffin, which is what we use to make a diagnosis, and also what we keep in the collection. That's about it. We don't have too much else. We do have some eyes that have remained in formalin, mainly because either we're not interested in processing them, or they're too big, or there's one other reason. We did get an example of what we're pretty sure is a blue whale eye, and it's massive. It's way too big to have in the collection. We did do an MRI scan on it so we can evaluate some of the anatomy. And if you're interested, if you do a Google search using those search terms, the best Christmas ever, and then put the word whale in at the end of that, and you'll get access to an NPR report about our whale eye. We also have some great white shark eyes, which are also huge, but that's not the reason why we didn't process them. We didn't process them because these are beached specimens, and we processed a couple of them, and they were just too rotten. Had too much. They were dead on the beach too long to be of any use. And then we have a few other eyes which we've kept in formalin and not processed. Feel free to interrupt me if there are questions as we go along. So this is basically what we have in the collection in the form of pie. Like I said, our main purpose is to be a veterinary diagnostic lab, being of service to veterinary ophthalmologists and helping them to better understand the appropriate diagnosis for their cases, and also to better understand the natural history of the eye disease as it develops in the animal. Because our main source of eyes in the collection are from practicing veterinarians, it's heavily skewed towards dog and cat. You can see dog by itself makes up almost 2/3 almost 3/4 of the collection, and the cat is another third or so of the collection. The other animal that we have lots of, but just a sliver in the pie chart, is horse eyes. So if we get an eye and it's neither a dog, cat, or a horse, then we designate that eye as exotic. So the yellow slice is all of our exotic eyes collected together. So this next slide is we take just that yellow exotics portion and make a pie chart out of it. And this shows broken down by phylogeny what type of animals we have the most of. And not too surprisingly, the largest part of our exotic collection would be mammals, followed by birds, followed by reptiles, and amphibians, not so much, but we have quite a few bony fish, and then just a sprinkling of cartilaginous fish, arthropods, and mollusks in our collection. So I'm going to run through just very briefly some of the types of research projects that we get involved in the different categories. And then what I'm going to do is I'm going to take this program down and screen share our database and screen share. And then after that, screen share our Kodachrome slide collection, and we can do some practice searches on that collection. So I'm going to just touch very, very briefly on some of the research material that I'm most proud of that we've done. And we'll start out with our investigations of spontaneous ocular diseases in animals. And again, feel free if any of this catches your interest and you're interested in asking a question, feel free to do that either in the chat or unmute yourself and ask a question. So one of the things that we're probably best known for is our discovery of a relationship between trauma and cancer in cat eyes. There are actually 3 forms of cancer that we think have this share this relationship. In some cases, the relationship to trauma is more pronounced and obvious than it is in some of the other ones. We call these tumors the post-traumatic sarcomas. And the 3 forms are what we call the spindle cell, which is the most common form of cancer, and then lymphoma, which is a cancer of lymphocytic origin, and then and that's the next most common type of disease. And then the least common is a tumor we call either osteosarcoma or chondrosarcoma. Osteo if the tumor is primarily made up of bone, chondro if the tumor is primarily made up of cartilage. And in fact, many of these tumors are a mixture of bone and cartilage. So we classify them all basically as the same category of disease. The spindle cell tumors, and you can see all of them, no matter what the category is, they're hard to distinguish from one another just by looking at them. They all make up a tumor which spreads in the eye by circumferential involvement of the eye. Basically, it fills up the entire inside of the globe, and in many instances, it extends beyond the globe into the other tissues around the globe. So the spindle cell tumors distinguish themselves because when you look at them, both either on the microscope or just look at them at the sliced eyeballs, they all have evidence of rupture of the lens capsule, which is another indication of trauma. These tumors develop in the small number of cases where we actually have a named traumatic event, and we know the time of when the trauma occurred in these cats. There's usually about a 3 to 7 year latency period between trauma and when the eye is removed. And so most commonly in these cats, we don't get a history of trauma, and all we get is a history of longstanding eye disease. Interestingly, a lot of them are really not suspected of having a tumor, even though the neoplasm basically fills up the entire eye. But it's an opaque eye. You can't see into the eye because the cornea is completely opaque. And it turns out that the cell type that becomes cancerous, or what we call neoplastic, in these spindle cell variant of post-traumatic sarcoma is in fact the lens epithelial cell, which is interesting in itself because in humans, there's an entire little niche of science devoted to the fact that the only cell type in the human body for which there are 0 cancers associated with is in fact the lens epithelial cell. And here in the cat, not only do we see this, but it's the second most common primary tumor of the cat eye. So then the lymphoma, again, you can see it has a similar distribution. There, I think you can see just by looking at the slides here that there's less likelihood of lens capsule rupture, although in most cases there is lens capsule rupture. And we don't necessarily, we're quite sure that the tumor doesn't arise from the lens epithelial cells, but probably arises by a malignant transformation of an already existing inflammatory disease in the eye. And cats do that in other parts of the body as well. There's a form of lymphoma, cancer of the lymphocytes that occurs in the intestine of cats that arises from a long-standing inflammatory disease of the intestines. And then the least studied, the one we've studied the least is the osteochondrosarcoma. And we don't really know, in these cases, in most cases, there's evidence of lens trauma and there's always evidence of long-standing eye disease. But the eye does contain what we call mesenchymal stem cells. So little rests of cells or individual cells in the eye that are capable of differentiating into a variety of different tissues. And we think that those cells become activated after trauma and participate in the formation of a tumor. So again, I'll invite questions if there are any, but I'll go on to other.
Well, Richard, Padma asked if a cat with eye cancer is found, can the eye be transplanted?
No, we actually worked on there are labs around the country that are working on the concept of transplanting of ocular tissue. The surgery involved is extremely challenging. We put in a grant to one of our surgeons put in a grant to participate in that study, but other labs were funded. So I don't really know very much about where that work is going. It's certainly not something that your run-of-the-mill veterinary ophthalmologist would be capable of doing. The eye is removed and put in formalin.
Thank you.
So another disease that I'm proud of the work that we've done is to better understand the natural history of what happens in dog glaucoma. Dogs get glaucoma much like humans do, but there are rather profound differences in the way glaucoma presents in dogs than in humans. We were able to use our lab and specimens submitted to the lab to demonstrate that glaucoma in dogs has an added perspective over what you see in humans, or for that matter, what you see in cats or most other animals. And that is that the blood vessels that supply both the optic nerve and the retina actually get squashed, and those tissues lose their perfusion from the blood vessels. One of the ways we did that is to look at the optic nerve tissue and cut it, not in the way we usually cut it, but cut it directly across the circle of the optic nerve. And then these little structures up here are the blood vessels that not only provide nutrients for the optic nerve itself, but also nutrients for the inner portion of, the outer portion of the retina. And it's with techniques like this that we were able to demonstrate that the major blood supply, both to the optic nerve tissue itself and to the outer portion of the retina, is compromised in dogs that have glaucoma. And this is just an example. Yes.
Rich, we have a question from Rich in terms of the glaucoma. And he says that it seems that in veterinary medicine, no intervention is taken until the individual is believed to be completely blind. Is there anything that can be done preemptively?
Oh, that's a great question. Veterinary ophthalmologists are not able to sit a dog down and show it an eye chart and to have them read off the numbers. The evaluation of vision itself in the context of veterinary ophthalmology is very difficult. So the tests that we do to assess if a dog can see is to there are three things. One is you can shine a bright light suddenly in the eye, and the dog will squint. So that measures the presence or absence of vision at a level which in people we would say is totally blind, although able to see light. And then the next thing is to, not with a bright light, but to make a menacing move toward the eye, like suddenly moving your hand or something else toward the eye, and the dog will flinch if it's able to see that. So that is the equivalent in human of being able to read the largest letter on the eye chart, which is certainly would disqualify someone from human from driving. And then the next test would be to take something like a cotton ball and drop it, and to see if the dog will track that, or if the dog will follow that cotton ball. Not all dogs can be encouraged to be interested in that, so it's kind of hit or miss. But that's tests a little bit more fine vision. Still probably not quality of vision that would be required to be able to legally drive. And then if you want to quantify your vision, then you can have the animal walk through a maze, or choose a path when there are obstacles in front of it. So our ability to assess vision itself is extremely poor. And yes, by the time an animal presents for glaucoma, oftentimes because the disease is so profound, they're oftentimes blind at the first presentation, although that's not always those are the cases that we're likely to get the eyes in formalin. Yeah, but I like to say the veterinary ophthalmologists don't deal that much with vision. They deal with appearance of the eye, pain in the eye, and vision. If in fact the vision loss is so profound that the animal is bumping into things or things like that. I hope that answers your question. I hope you're not too disappointed with the practice of veterinary ophthalmology.
Thank you.
So this is just an example of any one of numerous little studies that we do to better characterize diseases of the eye. And this is a little study in which we followed the natural history of dogs that develop squamous cell carcinoma. That's a cancer of the surface of the cornea, or the surface of the skin throughout the body, and when it occurs only on the cornea, which is unusual. And so one of the things we discovered, although it was no surprise to us, was that what we call brachiocephalic breeds, dogs that have a smushed in face and have pooched out eyes, are more prone to this disease. So if you look at the pie chart here, the breeds that get this disease, the pugs, far and away the most common, English Bulldog, other forms of Bulldog, Shih Tzu, and then 34 other breeds, but many of these breeds will also be smushed in face breeds of dogs. In addition, virtually all of these dogs will have some form or another of exposure disease of their corneas. Think back to the configuration of the head, and you can make a guess as to why they have exposure inflammatory disease of their corneas. And then another feature which I'm not quite so sure about, because it's very difficult for us to get an absolute count of the history of this, but there are anti-inflammatory drugs that are commonly used in these dogs. And I have a strong suspicion that the use of some of these anti-inflammatory drugs is also a combined feature in dogs that develop this cancer. The good news is it's a very superficial cancer. It can be removed by a surgical procedure to remove just the front portion of the cornea, and that's usually the end of the problem. Yeah. Again, let me know if there are questions. And then another disease that we studied that was kind of interesting, and this occurs in even fewer. I should say that it's extremely common for pugs or bulldogs to develop chronic inflammatory disease of the corneal surface, and only a tiny, minuscule proportion of those end up getting this squamous cell carcinoma. So I'm by no means saying that if you have a dog that's receiving anti-inflammatory treatment to make it more comfortable with its eye disease, don't go screaming to your veterinary ophthalmologist saying that he's likely to get cancer, because the likelihood of that is extremely rare. And this is a disease that's also extremely rare, but probably also related to immune suppression of the cornea, and that is the infection of the cornea by organisms that you wouldn't normally think would be involved in that, and that is these protozoal organisms. This particular dog was from Missouri, and it has leishmaniasis. Another disease that we've seen is toxoplasmosis, and trypanosomiasis is another potential possibility for some of these dogs that develop corneal infections, aberrant corneal infections associated with immune suppression of their cornea. Interestingly, when a dog gets, so let's say, leishmaniasis or toxoplasmosis in a more typical organ, they usually mount a systemic immune response, and you can recognize the presence of the disease by studying their antibody levels. But when they get this disease entirely limited to the cornea, they don't have that systemic immune response, and we have to diagnose the disease by looking at it under the microscope and then sending it off to a laboratory that can specifically identify the parasite that's involved. So moving on to comparative anatomy, this is a study that we did a couple of years ago. Alicia was a veterinary student at the time. She's now a pathology trainee at Georgia, but extremely interested in wildlife diseases. And Lauren Henley is an evolutionary biologist. And so we used our collection of eyes from different animals, and we concentrated on hoofstock, even-toed ungulates and odd-toed ungulates. And these are animals, a lot of these animals, not all of them, but a lot of them live in the open. They don't live in areas where there's a lot of shade, and they need some sort of protection so, that would help to prevent the sunlight coming from above. This would be the upside of this horse eye. The sunlight coming from above from getting into their eye, just in the same way you would wear a baseball cap if you're out in the sun and you don't want a lot of sun to get in your eyes. This structure is known as the granular iridica. In horses, it has a special name, the corpora nigra. So we looked at animals from hoofstock and to see if they had a granular iridica or not, and then we used a specific database to try and get an estimate of what their habitat was, and we made an estimate as to whether or not they lived in an area that was a lot of shade or a lack of access to shade. And we showed that there was a relationship between animals that live where they have access to shade or don't have access to shade as to whether or not they have a granular iridica. And there was, of course, also a relationship to their phylogeny, to their closest relative, but both features were definitely in play in the development of a corpora nigra. I mean, a granular iridica. So this is an interesting study. There's a lab at Purdue that studies vision in animals, and one of the things that they do very commonly is to take a species of animals that they're going to study, and they have access to the eyes, and remove the retina, just the retina, and lay the retina flat on a microscope, and then just look straight on at the retina through a microscope. And with a special stain, then what they're mainly interested in is to look at where most of the ganglion cells, the cells in the inner part of the retina that define where information is being sent to the optic nerve and then the brain. And then they make a topographical map, and it gives you an idea of how if those animals have a specialized area of their retina that's specially designed for high degree of visual acuity. They didn't discover it, but already known is that many animals, birds, reptiles, fish, amphibians, have oil droplets in their retina, and the oil droplets are colored so that the color of oil droplet will define the color that particular photoreceptor, that particular cell, can deal with to make a signal to the brain. And the oil droplets that you usually find are easily seen here. I'm going to point to a few of them. Here's one, here's one, here's one, here's one, here's one, here's one, here's one. The different colors are not that obvious on this specimen. That's because the red sort of captures your attention. And this is the first time these little conical shaped structures were discovered in the eye. This is from a fly-catching bird. So they sent us some tissues, and we looked at these tissues under electron microscopy, and this is what we saw that corresponds to these red conical structures. This in the middle is a gigantic mitochondria. Now the mitochondria is responsible for energy production in the cell, and then we think that around it, these little white areas here are the red are the structures that contain the red material. And the reason why an animal would have a huge mitochondria is because it provides evidence that that particular cell has an exaggerated demand for oxygen, an exaggerated metabolic load. And one of the features of a bird that allows a bird to respond to visual stimulus with such incredible quickness, reflex wise, is that they're able to once the cone has been exposed to something, they're able to regenerate that ability of the cone to expose to something else extremely quickly. And the faster the reflex time, the more energy that requires. So we think these fly-catching birds, which have an incredible ability to pick up a fly right out of the air, needs to have this ability to regenerate these particular cones extremely rapidly. That's not proven yet, but that's the hypothesis that we're working on. And it won't be us; it'll be the lab at Purdue that continues to work on that. So this is a kind of a fun project that we did. We don't necessarily think of clams as having eyes. This is a clam called the disco clam. And if you want to know why it's called the disco clam, look it up on a YouTube video. The disco clam has a reflective band that, if you shine light on it, produces a sort of a disco-like light show. So a scientist was studying these, and they wanted to ask us the question: do the eyes in this disco clam function at a degree to which the clam itself could probably recognize this reflective signal from that band of tissue? So this is what we found. We looked at this. I won't be able to show you the eyes in the clam, but they have maybe 100 of these. Right in the mantle, in this part here. These are little tentacles. The eyes are not on the tentacles, but they're between the tentacles. And so this is what the eye looks like. This is basically the retina. The photoreceptive elements of the eye are on the inside here. And then there's a clear area, which you could say is analogous to the cornea. There's even some cells that may aggregate light toward the retina, or may not. We're not at all sure about that. So our conclusion was that no, this eye is not able to. This eye would be able to perhaps to recognize light, or perhaps be able to recognize shadows. The only thing this animal really has to do is open or shut. And if there's perhaps a big shadow coming towards it, it might inform this animal that it would be a good idea to shut. And we do not think this eye is capable of forming an image. And this is not so much a study, but we work closely, and you can the quality of this image is not very good. But Ivan Schwab is an MD ophthalmologist who studies whose specialty is the cornea, but his passion is comparative anatomy and evolution of the eye. And he asked us to participate in accumulating images for this particular book, which I think is a wonderful book, especially for those people interested in comparative anatomy. So he sent us interesting specimens, and then we provided him with microscope images of what those eyes look like. I call him the Indiana Jones of comparative anatomy, because he would go around the country or the world for people that had interesting eyes that he wanted to evaluate, and then he would send us the specimens. And then the last part of type of research we did is to participate with drug companies in drug development. This particular study, they were looking at the development of an eye that would be used in very young people. And so the studies were done on very young dogs. And we discovered a peculiarity of the retina that wasn't related to drug use at all. And so we think it's a development stage in the normal maturation of the canine retina. So that's what that publication is all about. And this is probably a very important series of studies. We did a number of studies, and this is monkey eye, and it's a model of age-related macular degeneration where laser burns are produced in the back of the eye. And those laser burns, when you study them morphologically, have a lot of the features that age-related macular degeneration. And they discovered that they could modulate this. They could decrease the significance of this by using drugs that combat against the formation of blood vessels. And this treatment is now what's used from 3 different pharmaceutical companies as a standard of treatment for age-related macular degeneration, injections into the eye of these types of drugs. We played an important role in the development of those diseases. So my next, let me just check on the time here. Ooh, do I have time?
Yep, let's go ahead.
You sure?
Yep. Any questions so far right now from the group? All right, let's go on.
Okay, so this is what our database looks like. This database has a number of different pages. The specimen page is where we put information from the referring veterinarian, and then we'll choose a breed of animal and a species of animal, and then the client picker is where we log in the name of the clients, and then the species picker, I'm sorry, is where we define the species. And then the page that I deal with most is the medical page, and that's where we have we log the diagnosis, and we do it in 2 different ways. So one is directly off the report, and we have a student that transcribes directly copy and paste from the report into the database here, and then I personally enter what's called the DX, or the short diagnosis. In both instances, we don't code our diagnoses, but if you're going to have a useful collection and use terms which will allow us to search for the things we want to search for, you have to be very careful that you don't use different terms for the same process as you go along. So I try and drum into the people that are making the diagnosis to use very specific terms, and then when I'm putting these diagnoses in, I will check and make sure that the terms I want to be in this part of the diagnosis are up here as well, which allows us to search. So let's do a couple of searches, and we're going to go up here and we're going to hit the find button, and then I'm going to put let's put cat in, and we're going to look for those post-traumatic sarcomas. So first I'm going to put an X here which will identify we're looking for a neoplasm. And then I know that we put the word PTS for post-traumatic sarcoma in for all of these cases. And then I'm going to put the word spindle in, because I particularly want to look at I want this search to be for the spindle cell tumors. So here we have 297 cases of this in the database. So now we're going to do some sorting, and these green buttons up here allow us to sort. So let's sort by breed. We have to answer a couple questions. And then we're going to go over here to the Reuter, and we're going to look at the summary. And let's move this to the center, and then let's expand it a little bit. So it's never very interesting, and the breeds that we see in cats are never very interesting because we don't get that many that are purebred cats, and most of our specimens are domestic short-haired cats. So there's nothing particularly surprising here. Now let's quickly toggle to the age, and here's this is the age here. Notice that the integers are the ones that have the most number of cases. So 5 years has 6, 7 years has 11, 8 years has 16, and then the numbers in between are in-between numbers. So here, 10 years, there's 27, 12 years, there's 27, 13 years, there's 30, and then the numbers start going down as the cats get older and older. They go up to 20 years of age. So it's largely a disease of old cats. And that's interesting to me because when we actually get a designation of when the trauma occurs, the trauma doesn't usually occur in old cats. And I have a suspicion, we don't have enough numbers to prove it, but I have a suspicion that that in fact, the trauma in these cats occurs when the cat is not old, but actually pretty young. Now I've lost my cursor. Hello.
Yeah, I don't see the cursor either. You might have to, yeah.
Now I see it. And I'm going to go back to screen share. Go back to you. Now I got it. So let's go then to eye, left eye or right eye. So 48% in the right eye, 43% in the left eye, 2% in both eyes. And then let's go to the sex. And now we're going to see that about 2/3 of the animals are males. And that we think is a feature of the cats that are most likely to have trauma to the eye. If you look at the breakdown of by sex of other diseases related to trauma in the eye, it's about the same. These pesky males are more likely to get into battles with other cats, and probably a cat scratch is the #1 cause for the initial traumatic event. So that's how we put together information about how these animals look. Let's look for let's do a exotic search. So let's do a find again. Let's go back to medical, and let's put the word. No, let's go back to here. Let's put in. No, I have to be sure that the word will be in here. Let's do pinniped. Pinnipeds are seals and walruses and sea lions, etc. So we have 285, or I guess that's 65 examples of pinniped eyes. So now we're going to sort by not by breed, but by species. We're going to see what those made up of. Go to our summary. And so this will give you a breakdown of the different species of pinnipeds that we have in the collection. So you get an idea of how we can facilitate the use of our collection for research purposes, both research of spontaneous animal diseases and also research of comparative anatomy. Now I'm going to very quickly show you our image collection, and we'll do some of the same. That's interesting. So we have 31,000 accessions into our image collection. And this is, you can get an idea what most of them look like. We also collect images of the people that come into the lab. But this is just scrolling through. These are from most recent to most distant. Now let's try a search for this as well. So we need to go to keywords. Here, I wish this was I wish that it was automatically on keywords, because that's about the only one I use. Then I'm going to put in contains, and I'm going to do again cat, and we're going to put in tumor, and I'm going to put in PTS, post-traumatic sarcoma again, and we're going to put in spindle. No, I have to go not with begins with, but contains spindle. We can put together 200 and I think that says 96 examples of images of the spindle cell post-traumatic sarcoma in cats. We can modify this further. If we don't want to look at the gross specimens, we want to look at microscopic sections. Now we're just looking at specimens from the microscope, through the microscope. So I hope that gives you an idea of the type of work we do, the way we use our collection. And again, once again, the main thing that we're most proud of is our achievements in clarifying the natural history of spontaneous eye diseases mainly in dogs and cats, but horses as well, and then to a lesser extent exotic species. We're interested in not only comparative anatomy, but we're also interested in the pathology of. I chose pinnipeds. One of the reasons is actually the California sea lion is one of the species that we have many specimens of, and we've been a participant in helping to study the disease categories of the sea lion eye. So I'm going to end here. If there, let me know if there are questions. I'll try it.
Yeah, thank you, Dr. Dubielzig. This was phenomenal. Go ahead and start putting your questions in the chat, or you can raise your hand and I'll call on you. One question: when you were looking at the database, is that available to the public, or is that just something that's internal?
No, the file is called FileMaker Pro, and one of the quirks of FileMaker Pro is there's no save mode. If you change something or if you add something, it stays. So we do not want anybody but people in the lab having access to that.
And is your lab the only one that is collecting this kind of data, or others doing that? And then is there a way to combine those data sets?
We're definitely not the only one. I think we're the best organized and far and away the largest. I haven't. We do on occasion when we're studying an unusual or rare disease, and we know another lab has some examples of them. We will combine our caseloads with another lab, but under those circumstances, they're they're probably not relying on accurate searches from their database. They're relying on using their memory for these unusual cases and putting together a small case series.
And you mentioned that some of the beached animals, or an animal that's been dead for a certain period of time, the eyes are unusable in terms of the collecting the data. Is there a time? Is it time and temperature? I mean, when the eyes are still usable for collection?
Time and temperature would be the two most important features. And I'm unable to say what the amount of time is. Even within a matter of minutes or hours, the degeneration of the retina begins, but the eyes are still quite usable for probably several days. In most of our specimens, these are eyes that are removed at surgery and plopped right into a fixative, so there really is no time whatsoever. We have a smaller number of eyes that have come from the postmortem room. That's an unusual way for an eye to get to us, but once again, medical autopsies are usually done in a timely manner, and the carcasses are preserved in a cool room. So usually those eyes are usable. It would be an animal that dies in the field and lays out in the sun for a long time, or in the case of our beached sea animals, get discovered lying on the beach. Probably animals that have died at sea and then washed up as dead weight onto the beach, and then sat there in the sun for a while.
So the exotic animals that you get, do they come from controlled environments like a zoo or something?
Most of our exotic animals, we had relationships with a number of zoos, particularly when I was in charge of the lab, because I would offer discounted or free service if they would send us specimens, and actually they'd send us more specimens that we could use, and then they would allow me to just triage section in the ones that I felt I needed. But most of those came from a zoo, and again, most of those are animals that went to their local pathologist for autopsy, and the eyes became available that way. That's the most common. We did also work with some biologists that had access to specimens, and then Ivan Schwab would oftentimes go either to aquariums or zoos, or basic scientists who are working with interesting specimens.
And what about when you're taking the slices? Do you just is one slice sufficient to get all of the data that you need from that particular eye, or do you take hundreds of slices from one eye?
That's a great question. Actually, at the medical school, they usually take a whole bunch of different slices. That of course costs money, and the amount of money, the amount that we can charge for our services compared to what they get charged through third payer programs in the medical school is minuscule. So we basically take one section for most of our mammals. We take what we call a vertical section. You can orient the eye so you can tell the top and bottom portion. And we try to make a section that includes the optic nerve, the middle of the lens, and then the pupil. And if we've got all of those things in the same plane, then pretty much we can assure that we've got a central section of the eye. That's what we aim for. If in fact there's something, and we'll make careful records when we're looking at the eye grossly and examining it. If there's something that won't fit in that customized sectioning mode, we'll either change the sectioning mode, make an oblique section or a horizontal section, or we will submit pieces that normally would be trimmed away, or we will first make the section that we're interested in, and then we'll instruct the histopathology lab to reorient the block, or either that or cut deeper into the block to sample a second area. So using those techniques, we're usually able to find most of the areas we're interested in. In looking at the exotic specimens, sometimes sometimes there's a good reason for characterizing the anatomy by making a completely different section, plane of the eye, and we will on occasion do that. Not usually until we have enough specimens that we can document the anatomy in the usual plane.
Great.
And then among animals that we see fairly regularly, when we get a primate eye, rather than a vertical section, we'll make a horizontal section, because that will give us the best chance of sampling the fovea, the part of the retina that's used for acute vision.
So Doris asks, some of the specimens seem to show retinal detachment. Is this due to fixation artifact or related to underlying ocular pathology?
Apparent retinal detachment is one of the most common artifacts that we see. Remember, I told you that the eye begins to degenerate within hours of when it's removed, and the main thing that we see is apparent artifactual detachment of the retina. We can use clues in the tissue to give us an estimation of whether that retinal separation is artifactual or not. It's not 100%, but it's pretty good.
And she also asks, is ocular melanoma frequent in animals?
Melanoma is the most common tumor of the eye in virtually every animal that I know that has enough tumors documented in the eye to have an idea about it. It's the most common in people. It's the most common in dogs, cats, horses. Beyond that, there aren't enough tumors documented in any particular kind of eye to say.
Another question is, what are you looking for in a beached animal?
The main reason would be to study the anatomy. But on occasion, there'll be some recognizable disease, or perhaps there'll be a whole bunch of animals that are beached, and maybe some of them will have recognizable eye disease. I don't think we've ever done a series like that, but that could be a possibility.
You mentioned before, everybody asks you what's your most interesting or favorite eye that you have in the collection, or most unusual. Can you share that with us?
Yeah, so I was asked that question. I should have been prepared for that question from anybody. It's sort of a no-brainer of a question. But I was asked that question by a little collection around a table of medical physicians who are eye pathologists. And I wasn't ready for the answer. I wanted to say that I had no favorites, that they were all interesting in their own way, but they were having none of that. So I had to choose one, and on the spot I chose the chameleon eye. Now, if you've ever looked at a chameleon, you know that the eyes work independently, so they move about independently. And the chameleon is predator, and it catches its prey with a tongue shoot. And I've never tried to catch an insect with my tongue, but I imagine it's not very easy. And I imagine you have to have a pretty good idea of how far you have to shoot your tongue in order to catch that insect. So the chameleon has a very interesting way of judging distances using their eyes. When they're just in their environment, their eyes are going all different ways. But when they're focused on an insect, they go together. But even under those circumstances, they're not using stereopsis. They're not using both eyes and then a combined image to judge distance. They're using each eye individually. And the way they do that, it would be the same way you'd use a microscope. They have their structure, the optics of their eye is such that they are able to produce a very large, almost like a telescope image on their retina. And the retina is really nothing more than one big fovea. They have no peripheral vision, which is another reason why they have to constantly be searching their environment. They're both predator and prey. So they're able to use their eye, the focusing apparatus of their eyes, to calculate the distance away that an insect is, and so somehow the brain transmits that information to the tongue. The tongue explodes outward because a circular muscle around the tongue tightens, and then releases a stop apparatus, and the liberated tongue shoots out in whatever direction it's pointing. So there are, of course, a number of other interesting eyes, but I chose the chameleon.
That's amazing. That's interesting. They have to be, predator and prey, and the eyes work that way. Doris asks, hooved animals have rectangular pupils. Is that explained by irises located on top and bottom only instead of circular granular iridica? So why in octopus?
Why the octopus, I'm not sure. So I explained that if you want to find a rectangular pupil, you want to go to an animal that lives in the prairie. Well, neither the shark nor the octopus qualify for that. So there must be another explanation. So there are people who study the eye from the point of view of optics. And I am not one of those people, but there are some optical advantages of having a pupil the way an octopus has. Octopus has a really interesting eye. Their retina is backwards to the way a vertebrate eye is, and they only have one color variant. So that although they have quite good vision, they only have black and white vision.
And do you do any with insect eyes, with the compound eyes?
I love insect eyes, and our association with Ivan Schwab has led us into looking at insect eyes. I know almost nothing about them, except I know a little bit about the anatomy using a regular microscope. And there are not very many people that study insect eyes that use regular standard paraffin embedded microscopy techniques to study the eyes. So maybe under those, because of that, we have a kind of a unique collection. Right now, Ivan Schwab is working on a project to more clearly define the way a jumping spider sees. You're familiar with a jumping spider that has great big eyes in the front. So a jumping, if you look at visual acuity relative to body size, the jumping spider has the best visual acuity relative to body size. And Ivan has sent us 2 different species of jumping spiders and wants us to get sections that highlight those big central eyes. He's already sent us one years ago, and we have, I think, a beautiful section of it, but I want to get even better sections. Since I'm not going in very often, it's kind of a slow process. I will help the histolab to cut into the eyes when I'm in there, and then they'll do their work, and then I'll come in maybe a few days later, and we're working our way through that paraffin block to try and get perfect sections of a jumping spider, of Ivan's jumping spider eyes.
Very cool. Any other questions for Dr. Dubielzig this evening? Yes, I agree. Very fascinating, Doris. Bob says he doesn't have any questions, but he thoroughly enjoyed the introduction to the lab's work and collections. I agree. Thank you so much for sharing your time, your expertise, giving us the inside look at the lab, the work that you did. And thank you and all of the folks in your lab for the incredible work that you're doing in ocular pathology. It's really a benefit to our collective knowledge in the world. Thank you so much.
So if there's anybody out there in the audience that's really interested in this and wants to have a day-by-day experience in comparative anatomy or comparative pathology, I do run a Facebook page. You can probably find it by searching for the Comparative Ocular Pathology Laboratory of Wisconsin. I don't think you can find it by searching COPLOW, because Facebook doesn't allow acronyms in their title, but you can probably find, or just put in COPLOW in Google, and our homepage will allow a click to our Facebook page. And if you like it, Facebook page is basically nothing more than 2 pictures every workday, either pathology or comparative anatomy.
Well, that's great. If you can't get enough of the eyes, make sure that you like and follow the lab.
I'm interested in those people that can't get enough of the eyes.
Right. And I know that I have a new perspective, and I'll be looking at the world with new eyes because of what you've shared with us this evening. So thank you very much. Everybody, stay well, stay healthy, stay curious, and I hope to see y'all soon. Another Thursday next week is urban stream restoration, and then antlers, amazing antlers. So make sure that you've RSVP'd for those, and we'll see you soon. Take care, everybody.
Bye. Thanks for having me.
Thanks so much.